Hi everyone, welcome to the PowerPoint for Chapter 1, An Invisible World. The first question is, what is microbiology? Microbiology is the study of all living organisms that are too small to be visible with the naked eye. This includes bacteria, which will be the main focus for our semester, especially with regards to lab, as well as archaea, viruses and prions, which are both considered non-living, fungi, protozoa, and algae, all collectively known as microbes. These microbes play key roles in things like nutrient cycling, biodegradation and biodeterioration, climate change, food spoilage, the cause and control of disease, both human and otherwise, and biotechnology. Fermented foods and beverages also require a process known as microbial fermentation, which uses bacteria, mold, and/or yeast to convert sugars to alcohol, gases, and organic acids. Figure 1.3 from the first chapter of OpenStax shows a microscopic view of Saccharomyces cerevisiae, the yeast responsible for making bread rise, as seen at left. Yeast is a microorganism. Its cells metabolize the carbohydrate in flour, which is seen in the middle panel here, and produces carbon dioxide bubbles, which causes the bread to rise, as seen at right. Here we have a table from Chapter 1, which shows the various foods and beverages, as well as other products produced through the contribution of microorganisms. And as you can see, this includes popular foods such as cheese, alcoholic beverages, things like soy sauce and vinegar, yogurt, sour cream, artificial sweeteners even, bread, as well as things like antibiotics, human growth hormone, human insulin, the enzymes that are found in laundry detergents and dish soap, so on and so forth. Figure 1.1 from the OpenStax text shows a veterinarian getting ready to clean a sea turtle that's been covered in oil following the Deepwater Horizon oil spill in the Gulf of Mexico back in 2010. After this spill, the population of a naturally occurring oil-eating marine bacterium called Alcanivorax borchomenis skyrocketed, helping to get rid of the oil. Scientists are working on ways to genetically engineer this bacterium to be even more efficient in cleaning up future oil spills. Now, if we look all the way back in history, pretty much as far back as we can, we see that several ancient civilizations appear to have had some sort of understanding that disease could be transmitted by things that could not be seen with the naked eye. So in panel A, we have something called the cloaca maxima, or greatest sewer. And that's the part that's boxed out in red up in the upper left-hand panel. Now, this cloaca maxima, and keep in mind that cloaca is the singular opening both for urine and feces found in birds and other such organisms. So it kind of makes sense that that is also a name that equates to greatest sewer, right? Waste removal. And this ran through ancient Rome. It served as one of the world's earliest type of sewer systems. So it was constructed in ancient Rome in order to drain local marshes and remove the waste of one of the world's most populous cities. And it carried this effluent to the river Tiber, which then ran beside the city of Rome. And in panel B, down below, we see these ancient latrines, these toilets, right, that emptied also into the cloaca maxima. So the thought behind this is that even in early times, such as in ancient Rome, there was still some sort of wherewithal that we had to separate out, even if we couldn't see them, some of these what would in the future become known as microorganisms, because there was at least a crude knowledge that it could make us ill. Figure 1.5 shows three historical figures who contributed largely to the field of medicine at large. So all the way at left in panel A is Hippocrates, who is known as the father of Western medicine, and he believed that diseases had natural, not supernatural or religious causes. In the middle is Thucydides, and he is considered the father of scientific history, and he advocated for evidence-based analysis of cause and effect reasoning. He observed that survivors of the Athenian plague were subsequently immune to future infection. So if you think back all the way to 460 to 395 BC, there was still this notion of immunity. And then all the way at right in panel C is Marcus Terentius Varro, who proposed that disease could be caused by, quote, certain minute creatures which cannot be seen by the naked eye. While ancient peoples may have suspected the existence of invisible, quote, minute creatures, it wasn't until the invention of the microscope that their existence, meaning the existence of these proposed minute creatures, could actually be confirmed. While it is unclear who exactly invented the microscope, the Dutch cloth merchant Anton van Leeuwenhoek, who lived between 1632 and 1723, was the first to develop a lens powerful enough to view microbes. Nearly 200 years after van Leeuwenhoek, the golden age of microbiology spawned a host of new discoveries between the years 1857 and 1914. Figure 1.6 shows two individuals whose contributions were absolutely paramount to the field of microbiology in particular. The first, and probably a name that's not unfamiliar to many, shown at left in panel A, is Louis Pasteur. Louis Pasteur is credited with numerous innovations that advanced the fields of microbiology and immunology. He demonstrated that fermentation is caused by microorganisms. He also invented the process known as pasteurization, and he helped to develop vaccines. The man pictured at right in panel B is Robert Koch, and he identified the specific microbes that cause anthrax, cholera, and tuberculosis. He was also the first to demonstrate the connection between a single isolated microbe and a known human disease. The neck that you can see, so if anyone's ever participated in like glass blowing, maybe even in a chemistry class, as you heat glass, you can bend it. Okay, so what he did was he heated it and he made this S or swan-shaped neck to the flask. And that was key because he didn't have to plug the neck of the flask. It would allow in air, but the microbes would get trapped, as you can see in the lower left-hand panel. They would get trapped in that S curve. So this was huge because it showed physically that microbes were present. But if you prevented them from entering the broth, then the broth would not become contaminated. So basically he showed that yes, there are actual entities, although we might not be able to see them in small quantities, they're there. And that's what causes contamination of the broth, not just spontaneous generation. Here we take a closer look at the contributions of Robert Koch, specifically the formation of something called Koch's postulates. Basically, Koch's postulates was the first ever documentation, if you will, that proposed that one specific microorganism was the cause or was responsible for one particular pathogenic or disease state. So the first postulate states that the suspected pathogenic organism, the suspected microbe, should be present in all cases of the disease and absent from any healthy animals or individuals. Koch's postulate 2 was that the suspected organism should be grown in pure culture, meaning a culture that contains only that one microorganism and nothing else. 3, cells from that pure culture of the suspected causative microorganism should cause the same exact disease from the original organism from which it was isolated, and be able to cause that same disease state in an otherwise healthy organism. And then fourth, that organism should be able to be re-isolated from the newly sick animal and shown to be identical to the original microorganism that was originally isolated. Also involved in our initial discussion, just sort of setting the tone for this first lecture, which is just an introduction to everything that we're going to be going over this semester, is just a reminder. Many of you have, I'm sure, had this in previous science classes, but a discussion of taxonomy. Taxonomy is the classification, description, and identification and naming of living organisms. Classification is the practice of organizing organisms into different groups based on their shared characteristics. Now, taxonomy divided the world into three kingdoms: animal, plant, and mineral. Within the animal and plant kingdoms, the father of nomenclature, Carl Linnaeus, grouped organisms using a hierarchy of increasingly specific levels and sublevels. And he called the naming scheme that he came up with binomial nomenclature. Bi means two, nome is for name, right? So to name things so that they would be universally recognized, right? Not just like regional or nation-specific names, but rather all across the globe. He came up with a two-part naming system. So for those that may not be familiar or just need a quick refresher, these two-part names are always written in italics. The genus name, the first name, always has the first letter capitalized, and then the species or specific epithet is all lowercase. Phylogenies are evolutionary relationships of all the different species on Earth. So it's like a giant family tree, if you will. And in these tree-like or phylogenetic diagrams, groups of organisms are arranged by how closely related they are thought to be. In early phylogenetic trees, the relatedness of organisms was based on their visible similarities. Today, relatedness also includes things that we can't see, such as genetic, biochemical, and embryological comparisons. Ernst Haeckel, he is one of the first people back in 1866 to render a tree of life. So he published a book called The General Morphology of Organisms, and this book contained three kingdoms: Plantae, Protista, and Animalia. He later added a fourth kingdom, Monera, that included unicellular organisms that lacked a nucleus, which we find out later have become the prokaryotes. So in figure 1.10 of your OpenStax book, we see that nearly 100 years later, in 1969, American ecologist Robert Whittaker proposed adding another kingdom, fungi. He also contained a level above the kingdom level, the empire or super kingdom level, in order to distinguish between organisms that did have membrane-bound nuclei, what we now know as the eukaryotes, and those that do not, the prokaryotes. Even today, the taxonomy of living organisms is continually being reevaluated and tweaked and refined as we have further and further advances in molecular biology. Now, that brings to light the role of genetics in modern taxonomy, because, even 20, 30 years ago, we didn't have these sophisticated genetic analysis techniques that we have today. So genetic methods allow for a standardized way to compare all living organisms without relying on observable characteristics that can often be subjective, right? Like shape of a leaf or color. Rather, genetics is sort of hard and fast, right? It's the sequence of A's, T's, C's, G's, and in the case of RNA, of U's as well. Modern taxonomy relies heavily on comparing the nucleic acids, DNA or RNA, or even proteins, right? The end product of DNA and RNA from different organisms. In the 1970s, American microbiologist Carl Woese discovered what appeared to be a living record of the evolution of organisms. He and his collaborator, George Fox, created a genetics-based tree of life that was based on similarities and differences they observed in the gene sequences coding for what are known as small subunit ribosomal RNA, or rRNA, of different organisms. If any of you can recall back to your general biology, maybe even in high school, the little factories in cells, whether they're prokaryotic like bacteria or eukaryotic like us, have these things called ribosomes. And part of what makes up the ribosome is that rRNA. In the process of doing this, Fox and Woese discovered that a certain type of bacteria called archaebacteria, now what we know as archaea, were significantly different from other bacteria and especially from eukaryotes in terms of their very small rRNA gene sequences. So to accommodate this difference, Woese and Fox created a phylogenetic tree with 3 domains above the kingdom level. And these are the domains that we still have today. And these include archaea, bacteria, and eukarya. And we're going to go into more detail on everything that we've talked about today as we proceed through the semester. Now, as far as naming microbes, taxonomic names in the 18th through the 20th centuries were typically derived from Latin, since that was the most common language used by scientists when taxonomic systems were first created. Today, newly discovered organisms can be given names derived from Latin, Greek, or English. Sometimes these names reflect some distinctive trait of the organism. In other cases, microorganisms are named after the scientists who discovered them. So, for example, E. coli was discovered by Theodore Escherichia. So the E is for Escherichia, and then the coli part is because this bacterium affects the colon. Figure 1.12 simply shows the relative sizes of various microscopic and non-microscopic objects. Note that a typical virus, for example, measures about 100 nanometers, a nanometer being a billionth of a meter. This is 10 times smaller than a typical bacterium, which is usually about 1/1,000,000th of a meter. Okay? And in turn, a typical bacterium is at least 10 times smaller, than a typical plant or animal cell. So it gets to the point where beyond a bacterium, we can't see any other microorganisms with a regular light microscope. For that, for something like a virus, we would need an electron microscope. Now, prokaryotic microorganisms, just to reinforce, again, we're going to have more detail on this as the semester goes on, but prokaryote means these organisms still have DNA, but it is not housed in a nuclear membrane. So, prokaryotic organisms include both bacteria and archaea. Bacteria are found in nearly every habitat on Earth, including within and on the surface of human beings. Most bacteria are harmless or even helpful, but some are pathogenic, meaning disease-causing. A disease-causing microbe is referred to as a pathogen. And as I've reiterated before, in prokaryotic microorganisms, the DNA is not housed within a nuclear membrane, but rather just sort of hovers within a general region of the cell. Most prokaryotic microorganisms have cell walls, and if they're bacteria, the cell walls contain a polysaccharide known as peptidoglycan. Now, microbes are often described in terms of their general shape, aka morphology. and they have a wide range of metabolic capabilities and can grow in a wide range of environments. So the general shapes you see, and they're also included on the next slide, are coccus or spherical, bacillus or rod-shaped, vibrio or curved rod, coccobacillus, which is sort of a hybrid between a sphere and a rod. I say it reminds me of like a tic-tac. And then spirillum and spirochete. Spirillum is sort of like a worm, and a spirochete is like a corkscrew. Here on slide 20, we have again a reiteration of what I've gone over just on the previous slide, but also what you can see is there's more descriptive ways. So not only is a cocci bacterium spherical, but you can have diplococci, right, two side by side. You can have streptococci, which strepto means a chain. You can have a tetrad. You could have staphylococci, which is a pile of those cocci bacterium. So we learn as we go on, we can attribute certain characteristics to these bacteria simply through their name. Archaea are the other type of prokaryotic microorganism. They are also unicellular, but they differ from bacteria in their evolutionary origins, in their genetics, in their metabolic pathways, and in the composition of their cell walls and membranes. Unlike most bacteria, archaeal cell walls do not contain peptidoglycan, but instead they contain something called pseudopeptidoglycan or pseudomurine. Murine means wall. Like bacteria, archaea are found in nearly every habitat on Earth, even extreme environments that are very cold, very hot, very basic, or very acidic. Some archaea live in the human body, but none have been shown to be human pathogens. And so the name archaea, if you just think about it, and we'll talk about this the first Zoom that we have, should elicit something in terms of the word archaic, right? Which archaic means from the past. So archaeal organisms are believed to have been some of the first organisms ever to populate the earth. And if you know anything about the early conditions of the earth, they were not like they are today, right? It was dark. There was very little to no oxygen. There was very little to no sunlight. So it's believed that these archaeal organisms live in these quite extreme environments because that was the environment from which they originally evolved. So here we see a comparison in terms of attributes between archaea, bacteria, and eukarya. Archaea are prokaryotes and share certain traits with both bacteria and some with eukaryotes. Some archaea live in extreme environments and are called extremophiles. Extreme halophiles live in highly saline or salty environments. Halo means salt and phile means liking or loving. Now, most organisms, especially like bacteria, right? If you think about why in the grocery store things like canned sardines or even like a lot of food with a lot of preservatives in them, high salt, high sugar, don't need to be refrigerated. Usually high salt is antimicrobial, but certain organisms like halophilic archaea or bacteria thrive in these salty environments. Extreme thermophiles are organisms that live in very, very hot environments that would normally kill other organisms. And then finally we have the domain eukarya, which contains all eukaryotes, including both uni and multicellular, such as the protists, fungi, plants, and animals and humans. The major defining characteristic of eukaryotes is that their cells have a nuclear membrane or envelope around their DNA. Protists are an informal grouping of eukaryotes that are not plants, animals, or fungi. Algae and protozoa are examples of protists. Algae can be either unicellular or multicellular and can vary widely in size, appearance, and habitat. Their cells are surrounded by cell walls made of cellulose, a type of carbohydrate. You're familiar with cellulose as humans' dietary fiber. It's very abundant in things like celery. Algae are also photosynthetic, so they can extract energy from the sun and release oxygen and carbohydrates into their environment. They're producers. Protozoa are protists that make up the backbone of many food webs by providing nutrients for other more developed organisms. The protozoa are very diverse. Some move via flagella and some via pseudopods. Some are photosynthetic and some feed on organic material. Some are parasitic and some are free-living. Some are harmless and some are pathogenic. Giardia lamblia, for example, is a flagellated parasitic microorganism that colonizes and reproduces in the small intestine, causing a diarrheal condition known as giardias. Some multicellular fungi, such as mushrooms, resemble plants, but they are actually quite different. Other multicellular fungi are called molds. Molds are made up of long filaments called hyphae that form visible colonies. Fungi are not photosynthetic, and their cell walls are usually made out of chitin rather than cellulose. There are also more than 1,000 known species of unicellular fungi called yeasts. And what you see here on the lower left is a single-celled yeast called Candida albicans that is the causative agent of a condition known as thrush. I know when my oldest was born, he had pneumonia, so he had IV antibiotics, and his immune system was suppressed, and lo and behold, he ended up with this whitish covering all over his tongue. Helminths are multicellular parasitic worms that are not technically microorganisms in their full form because they're large enough in their full form to see without a microscope. However, they fall within the field of microbiology because diseases that are caused by them involve their microscopic egg and larval forms. So what you see at left in panel A is a beef tapeworm, Tinea sanguinata, which infects both cattle and humans. T. sanguinata eggs are microscopic, but adult worms, like the one that's shown here, can reach from between 4 to 10 meters, taking up residence in the digestive tract. In panel B, at right, we see an adult guinea worm, Dracunculus medinsis, and it's removed here through a lesion in the patient's skin by winding it around a matchstick. And if you notice, all the way at the right, I have put an image of the medical field sort of logo, the rod of Asclepius. And this actually came from what you see in panel B, right? The rod, that little like snake that you see, is actually a helminth, a parasitic worm. So that's where that symbol comes from. Now, viruses are acellular microorganisms, which means they are not composed of cells. They're not living. So essentially, a virus consists of proteins and genetic material, either DNA or RNA, but never both, that are inert, right, are completely inactive and cannot replicate outside of a host cell. However, by incorporating themselves into a host cell, they're able to sort of co-opt or take over the host cellular mechanisms in order to multiply and infect other host cells. And we'll have a whole lecture on viruses and prions. So microbiologists tend to specialize in one of several subfields. So bacteriology is the study of bacteria, mycology is the study of fungi, protozoology is the study of protozoa, parasitology is the study of helminths and other parasites, virology is the study of viruses, and immunology is the study of the immune system. And this is often included within the field of microbiology because the host-pathogen interactions are central to our understanding of infectious disease processes. Microbiologists can also specialize in certain areas of microbiology, such as clinical microbiology, environmental microbiology, applied or food microbiology. And this last figure figure 1.21 shows a virologist, right, one of these specialists, out in the field sampling eggs from a particular nest to look and see if they've been infected with the influenza A virus, which causes avian flu in birds. So that is it for the chapter 1 sort of introductory lecture. Okay, thank you for listening, and I will see you all on the weekly Zoom.